Hello and welcome to BMY 4302 Virology. This course is brought to you by Department of Microbiology, Faculty of Biotechnology and Biomolecular Sciences, University of Putra, Malaysia. I'm Dr. Eddie Chia. Today we are going to look at a group of viruses whose life cycle uses reverse transcriptase. Right, so basically what we are going to talk about at the beginning, we give you an introduction of this group of viruses. For example, their taxonomy, how they are being arranged, and what's the similar characteristics among this group of viruses. Then we'll look specifically into two groups of viruses, two families. One is Retroviridae, which is an RNA virus, and this is the families that where HIV is being classified. Then we use the second example, which is Hypanaviridae. It's a DNA virus, and this is where the hepatitis B virus are being classified into. So let's start with our introductions. Now, in the central dogma of molecular biology, we learned that it's the DNA, start from DNA, and then you have transcriptions go to mRNA or DNA to RNA. This process is called transcriptions, and then from mRNA go to protein translations. All right, so this is the central dogma of molecular biology, and we know that there is a step where the RNA can be reverse transcribed back to the DNA. So the process is called reverse transcriptions, and in order for this process to carry out, it needs a specific enzyme to carry out this process, which is called the reverse transcriptase, or in short, sometimes we put RT. All right, so this group of viruses use the reverse transcriptase in their replications of the genome. All right, so over here we can see the DNA to RNA. This is the normal transcriptions. So the RNA to DNA, this is the step where the reverse transcriptions happened. And for family Retroviridae, the reverse transcription happened at the very beginning, whereas for Hypanaviridae, the reverse transcription happened later in the life cycle. All right, so for the first two parts of these lectures, we're going to look at the Retroviridae, and then the second two parts, we'll look at the Hypanaviridae. All right, so now back to the taxonomy of virus. There are four realms of the virus, and one belongs to the RNA virus, which is Riboviria. Then we have another three which belongs to the DNA virus. Okay, so in this realm, Riboviria, we have two kingdoms. The first kingdom is Autonaviridae, Autonavirae, and then the other kingdom is Paranavirae. All right, so for the past few weeks, we have a look at the negative strain RNA and positive strain RNA. And there's another group of viruses that we didn't look at it, which is double-stranded RNA. So all these RNA viruses are really RNA virus, means that they don't have to go through the reverse transcribe to DNA. Their life cycle all depends on the RNA. So those group of viruses are being put under Autonavirae. So auto means right, correct, and para means side. So meaning that this is a true RNA virus, and this is like a partial RNA virus, because in their life cycle they have RNA and DNA part. So that's why Autonavirae, where all the RNA viruses are being located. Then Paranavirae is the reverse transcribed virus. So here, the kingdom have only one phylum, okay, and this phylum is Viricota. It's interesting that if you look carefully, because it's reverse transcriptions, and the scientists are trying to be creative, and so what you can see is REVTAR. So look at the class. The class is Retroviridae. So they only have one class, Retroviridae. All right, so this is the class. So reverse transcriptions. So they are trying to be creative, so they reverse the spelling, so it becomes ARTVER. So now you can see that the phylum is Viricota, and you know that it's actually the reverse spelling of Retroviridae. Revtra here. All right, so we have one phylum and one class, only one class, which is Revtra viridae. In this class, you have two orders. One order is Blueberviraris, okay, where this is where the family of Hypana-Viridae are being classified. So this is the only families for this particular order. Right? Hepatitis B is being classified into this family. Then the other family is Orthoviraris, and you can see again they are trying to be creative. Now this group of virus mainly they are looking at the retrovirus, which is the RNA, DNA, RNA part. So you can see it's actually reverse of retro, R-E-T-R-O, Orthoviraris. All right, so if you just remember the spelling, then it's easy. Orthoviraris, right? So this Orthoviraris have 5 families. So Bellpal-Viridae, Calimor-Viridae, Meta, Pseudo, and Retro-Viridae. So Retro-Viridae is the one that we're going to specifically looking at. And the other 4 families here, let's briefly look at it. So Bellpal-Viridae, there is only one genus in this group. Okay, the virus that classified in this group specifically target insects or invertebrate. So that is only the group of virus that classified in this group. The same go for Meta and Pseudo. These 2 virus group of families also infect insects as well as the fungus. So they infect yeast and so on. All right, so these are the 3 that infect insect. And then the interesting one would be Calimor-Viridae. So this is the virus that infects plants. Okay, Calimor-Viridae. So not only is it interesting in this aspect that it infects plants, but it also is a virus that doesn't have envelope, or the rest of them that have envelope. But this group of virus doesn't have envelope, and they are double-stranded double-stranded DNA as their genome. All right, so enough of this. Then let's look specifically on the Retroviridae. So the classifications of the final genetic trees that are being built to help us to classify in this group of viruses are using reverse transcriptase. So basically, because this group of viruses are so distinctive compared to the rest of the viruses, the reason being that they have this enzyme that couldn't be found or not found in any other viruses. So the scientists use the sequence of this enzyme, the amino acid or the DNA or the RNA, the sequence of this, and to analyze and to classify them into specific group. And what we can see here, these are the phylogenetic tree, and you can see Hypana-Viridae already out from the group, whereas the rest of them are still being classified in the same order, which is Orthoviraris. All right, so what we see here is Calimor-Viridae, Meta are closely related, and they are closely related to Retroviridae, whereas Bellpal-Viridae and Pseudo-Viridae are more closely related. All right, so even though Calimor-Viridae is double-stranded DNA virus and infect plants, you can see they are more closely related, meaning that they come from the same ancestor over the period of evolution. That's how they branch out. So what it means here is like we shouldn't just look at whether the genome is single-stranded or double-stranded or DNA or RNA. Sometime along the way, the evolutions happens, and this reverse transcriptase sequence will be more accurate to classify the virus into their respective order. Then Hypana-Viridae over here. Okay, so over here is the genomes of a virus, and you can see that they have specific genes that are being located and arrangement, and we'll look at it later. So these are the features that shared by these viruses. All right, like I mentioned, they have specific genes that, for example, the polymerase, presence of polymerase, presence of GAG stands for group-specific antigen. So AG is antigen, group-specific antigens. So, and then we have a long terminal repeat. This is something that we're going to look at later on. and how the priming is being carried out, how the replication, remember the replication needs primer in order for them to transcribe and so on. This is the priming mechanism. And then the genome types. All right, so if we look specifically into individual, and what we can see here, the presence of polymerase. In the polymerase gene, they coded for reverse transcriptase, RNase H, protease, and integrase. So basically, they coded for 3 or 4 genes in this case. So in most of the autovirus, what we see here is that all of them have RT, reverse transcriptase, RNase H, protease, and only the Colimor doesn't have integrase, but the rest of it have it. However, for Hypanaviridae, they only have reverse transcriptase. It doesn't have protease, doesn't have integrase. This is enough to separate the Hypanaviridae out from the group. Also, to look for the GAT gene, the group of group-specific antigens. So the common group of virus have the capsid or capsid protein or nucleocapsid. So in this group, all of them have it except for spuma, retro, virinae. But all of them have this, but Hypanaviridae, no. So not to say that Hypanaviridae doesn't have the capsid. They have a specific capsid, but this capsid is very distinctive compared to the rest of the retrovirus. So that is the reason why this Hypana is being classified in a separate order, okay? Bluebird Briaris. All right, so not only that, it also have a different priming mechanism that separate them. All right, so then you come back to like, why is Colimor viridae, even though it's double-stranded, as I mentioned earlier, even though Colimor viridae is a double-stranded DNA, and the rest of them are mostly RNA virus, why is it more related to the RNA virus than the DNA? So here it has the gene of polymerase. So you can see they are quite similar, and And the priming mechanism, this is the one that make the Colimor viridae more closely related to the rest of the family compared to Hypana, because the priming mechanism use tRNA as compared to terminal protein in Hypanaviridae, which we will look at it later on. So clearly, this double-stranded or DNA or RNA doesn't make the virus separate into different family. All right. So it's mainly just a type of the genome for the virus. So now we're going to focus on the Retroviridae. All right. So the Retroviridae gets its name from the retrograde of information, and this is the first when the virus are first being identified. So they give the name of a retrovirus, and that's give the name. So let us look at the structure of the retrovirus. So basically, what we have here there are 2 glycoproteins on the surface. So you have a glycoprotein that's responsible for binding. All right. So we SU them for surface. Okay, SU surface glycoprotein. Then you also have a transmembrane glycoprotein. This is the protein that has the part of the protein on the membrane. That's why it's called transmembrane, abbreviated as TM. Then underneath this glycoprotein, you have a layer of membrane. Okay, a layer of membrane that comes from the host. Then underneath the membrane, you have a layer of matrix protein, MA. Right, so this is the matrix protein, just like other negative RNA, influenza or NDV, they all have the matrix protein underneath the layer of membrane. Then moving inwards, what we see here is a capsid. All right, so this is the capsid. It's not icosahedral. It's a capsid just of different shape that surrounded the RNA. All right, surrounded the RNA of the virus. All right, so inside the capsid, we also have nucleocapsid. All right, nucleocapsid protein. This is the protein that binds to the RNA. So capsid surrounding it, nucleocapsid bind to the RNA. So inside the capsid, not only we have nucleocapsid, we also have the reverse transcriptase and integrase. Remember, the host will not have reverse transcriptase, and this enzyme is specific for the virus. That's why, like it or not, the virus have to carry their own RT into their virus. All right, so this is the basic structure of the virus. All right, so now in this Retroviridae, we have two subfamily: auto, mean true retrovirus, and spuma retrovirinae. So subfamily of auto retrovirinae, subfamily of spuma retrovirinae. All right, let's look at this spuma retroviridae. Now, this is a relatively new group of viruses that are being investigated. What is the distinctive difference between this group and this group of viruses is that the virus, for most of the, for all the auto retrovirinae, they use the cytoplasmic membrane as their membrane. Okay, cytoplasmic membrane during the budding. But for spuma retroviridae, they use endoplasmic reticulum membrane as their viral membrane. Okay, this is one of the distinctive characteristics. And also, the spuma retroviridae doesn't have nucleocapsid protein, the MP. Right? So this group of virus doesn't have that. So that classified them separately from there. Also, not only that, during classification analysis using phylogenetic tree, analyzing the RT, that helped them to separate this group and this group of viruses. So here we can see the genus. There are 5 genus here, genera here. Bovine means cow, cattle, bull, and so on. So bovine spuma virus, the virus that infect the cattle. Equine, horse. All right, feline, cat, and we have simian here. So these are the 2 that infect monkey, apes, and so on. So basically, these are the group of viruses that infect the animals. Then move on to subfamily of autoretrovirinae. So we have alpha, beta, gamma, delta, epsilon. So just like A, B, C, D, E. And then the last one would be the lentivirus. This is a specific naming of the group of virus. So of course, when you look at it, alpha, beta, gamma will be the simplest because it's A, B, C, the first, second, third. And then you have delta, epsilon, and lenti, which is the more complicated, the complex one. So phylogenetic tree analysis of this group of viruses using RT, we can see that all these group of viruses have been separated into their own branch. So alpha here, beta, gamma, circled by black box, meaning that they are simple virus, whereas you have delta, epsilon, and lenti, which is the complex, as well as the spuma, which are the complex virus. So over this, you can see like alpha, this is Rous sarcoma virus. You can see they are simple, there is no additional, meaning that whatever genes are present here is present in others. And also over the course of evolution, you can see new genes are being introduced into each of the branch that make them a separate group of virus in this case. All right, so let's briefly have a look at the viruses in this group. And what is the most interesting is that the first retrovirus that being analyzed will be the retrovirus that infect birds. So scientists found that there's this virus that causes cancers in the birds, and they start analyzing the virus. And over the course of a period of years, and a lot of findings come out from the research because the retrovirus is something really new. Remember, I mentioned the central dogma of molecular biology is always DNA to RNA and to protein, and it never go backwards. And that discovery of reverse transcriptase is the one that give a lot of insight of what this central dogma has to be more complicated compared to the simpler one. So because of this analysis of the reverse transcriptase, a lot of scientists that being awarded Nobel laureate because this is something really new. So over the times, and they only noticed that the retrovirus only infect birds, and they couldn't find any retrovirus that infect mammals until the first retrovirus that infect the primate, which is the primate T lymphotropic virus, or in short PTLV1. So what they say, from the name you can see, okay, in fact, primate, and it specifically target T cell, T lymphotropic T cell. So meaning that this virus specifically target the T cell and in the humans, and this is the first retrovirus that they found. And I'd like you to also look at the disease that caused by this group of retrovirus. You can see like mostly cause cancers or something that is quite serious, like you see lymphoma, all right, sarcoma, and so on, and neurological disease, anemia. So basically it just kills the cell or make the cell become cancerous. Also it can cause like immunodeficiency in humans or in cats, in monkey as well. So later during the applied virology, you will learn more about how the virus, the gene in this retrovirus can actually transform a normal cell to become a cancer cells. All right, so let's have a look at the genome organizations of the retrovirus. There are four genes, four group of genes that is very important. Okay, I'm not saying for, I specifically say for group of genes. So here, for group of genes, the first group of gene is GAG, group specific antigen that I mentioned. And then the second group is protease. The third group is polymerase, POL, polymerase. The fourth group will be the envelope, okay? So these are the four group of genes that coded for different protein. And these four group of genes are actually present in all the retrovirus. You see, you have alpha, beta, gamma, delta, epsilon, lenti. And what we can see from here, you have GAG, pro, pol, and end. So this is the simplest version of the retrovirus, the alpha retrovirus. You can see they have 4 genes, 4 genes, 4 genes, 4, and 4, and 4. Of course, as the virus become more and more complex, they come out with some accessory genes along the way. You can see ORF, TARX, REX, AB, and WIF, and these are so on. So as the virus evolved to become more complex virus, they have more accessory genes that are being included in their genome to make them to become more infectious or evolve to be, all right, to be a more great pathogens. Right, so the one that we are interested in will be the lentivirus. Okay, what's the point of learning the simple one? Let's look at the most complex one, which is the lentivirus. So in lentivirus, we have 10 species. You can see from this list. All right, bovine, caprine, okay, virus that infect cattle, goat, equine, horse, feline, cat, human, and so on. All right, so these are the group of viruses that being grouped under this lentivirus genus. All right, the one that we are interested in is these two species, which is HIV-1, human immunodeficiency virus 1 and 2. You can see these two, even though they are both called human immunodeficiency virus, they are not being grouped together. They are actually two separate genera. Right, let's look at the virus structures for this. Okay, well, from now on, I will just use HIV rather than human immunodeficiency virus. So the genus is lentivirus. There are two groups of HIV, HIV-1 and HIV-2, where both of these viruses can cause acquired can cause acquired immunodeficiency syndrome, or AIDS in short. All right. So not all the virus will eventually cause this. If the person who are infected are not undergo any treatments, so eventually the virus will make the immune system so low that they become immunodeficient. All right. So phylogenetic tree analysis of this group of virus using reverse transcriptase again. So it's actually separate the virus into two groups. What you can see here, SI means simian, so means that the virus, the simian immunodeficiency virus, this virus infects the monkeys and apes and so on. All right, so what we can see here is this is HIV-1, this is HIV-2. You can see that HIV-1 and HIV-2 in the phylogenetic tree already branched out earlier in the time. So meaning that HIV 1 and 2, they are, even though they are in the same genus, they are not closely related to each other. In fact, you can see from here, the simian virus here is more closely related to HIV 2 compared to HIV 1. All right? Or HIV 2 is more closely related to the simian virus here compared to HIV 1. So the same goes for HIV 1. You can see HIV 1s are being grouped more closely related to simian virus in this group. So what we can see from here is that we believe that HIV 1 is evolved from this group of simian virus, and this HIV 2 is evolved from this group of simian virus. So they have totally different virus that evolve, that become human immuno, HIV 2 and HIV 1. All right? So even though they are being named 1 and 2, it sounds like they are quite closely related. Just like hepatitis A virus and B virus and C virus, they are all different viruses. Okay, so hope you still remember paramyxo virus and Hipana virus, Viridae, and so on. All right, so what other differences that compare HIV 1 and HIV 2. So if we look at the virus replications in this case, you can see that HIV 1 replicate much faster compared to HIV 2. So higher replications, and this is lower replication, but it doesn't reduce the infectivity of the virus. Simply means that this virus replicate faster than this virus. So because it replicate faster, then you observe like pro-viral replication, you can see that they have higher viral RNA load, meaning that when the virus infects the cell, they replicate faster, and then of course when they replicate faster, you will have more RNA in the cell as compared to this one replicates slower. So it's quite straightforward and it's quite logical. And because they replicate faster, they produce more virus, they are more fit, 100-fold more fit compared to HIV 2. All right? So in terms of disease that caused by this, they still have the same kind of disease that being caused. Even though this replicate faster, this one replicate slower, but over the time it will balance out. All right. So over here, what we can see here is the viral load in semen. Okay, if the virus is present in the semen, what you can see here is that because it's replicate faster, of course you will expect this to be higher and lower in this case. Rate of evolution evolves slower, evolves faster, okay, because it takes longer time in the cell. Right. The other distinctive characteristic of these two viruses is like when the virus attached to the receptors, the virus need a receptor on the cell for it to attach to, and in HIV they have to have additional core receptors for them to enter the cell. For HIV-1, they have two, which is CCR5 and CXCR4. Whereas for HIV-2, not only it has a CCR5 and CXCR4, they also can use like CCR1, 2, 3, and CXCR6 as their core receptor, meaning that these have more core receptors that it can be used for it to enter the cell. So basically, this is some sort of a summary comparison between HIV-1 and 2. Not only genetically they are different, even the virus replications and infectivities is also slightly different and comparable compared to each other. So let's look specifically on the structure of this virus. Okay, as I mentioned earlier, the virus, on the surface of the virus, there are two glycoproteins. So, which is coded by N envelope gene, right? These two glycoproteins that on the surface of the virus is GP120 and GP41. So GP120, as I mentioned earlier, short abbreviated to SU or surface. And then GP41 is transmembrane, which is the one in blue. Transmembrane means that the protein crossed the membrane. That's why it's called transmembrane. So these are the two proteins that appear on the surface of the virus. All right. So the structure of this virus, the structure of this surface and the transmembrane protein, it looks as if like a mushroom. Okay. where this is the stem of the mushroom and this is the head of the mushroom. Right, this is the surface protein. So you also notice that there are other protein on this surface, but this is actually the HLA, human lymphocytic antigens. This HLA from the host, or MCH on the that coming from the host. So this is not part of the viral gene. All right, moving inwards, this is the membrane, and HIV get its viral membrane from the cytoplasmic membrane. Right, this is the membrane. Underneath this layer of membrane, you have matrix protein, which is in green color. All right, so the group-specific antigens is the genes that coded for 4 protein. Right, we have 1 GAT17, GAT24, GAT7, and GAT6. Okay, group-specific antigen. Right, 1, 2, 3, and 4. These are the 4 genes that coded by the group-specific antigen gene. So underneath here, we have the capsid, okay, or matrix protein, The green is the matrix protein, which is P17. Then moving inwards, the orange color, this is the capsid, P24. All right, P24. And inside the capsid, we have the RNA, and the RNA is surrounded by nucleocapsid protein, P7. All right, so outside of this capsid, the orange color is a layer of blue color, and this is the P6. All right, so now we have GAT gene that's coded for 4 protein, P6, P7, P24, and P17. These are the 4 protein that coded. So inside the capsid, we have the RNA, and bear in mind the RNA for HIV is a diploid. What it means that the virus carry 2 genomic RNA. These 2 genomic RNA is not double-stranded; they are individual. But these 2 RNA is actually identical. They are not complement to each other, but they are identical. All right, they are identical in the gene in the capsid. So why diploid? Okay, why must it be a diploid? Why must it has 2? So basically, 1 RNA is sufficient for the virus for replications. It's somehow going through evolutions, and you have 2 that enter the cell. All right. So inside, I mentioned that because the virus is a retrovirus, so this retrovirus need to carry their own reverse transcriptase, which is coded by polymerase gene. So in the polymerase gene, it coded for reverse transcriptase, which is present in the capsid. And then we also have another enzyme called integrase, which is coded by the polymerase gene as well, integrase, inside the capsid as well. All right, so we have covered the 4 gene, and we also have another protein, which is part of the polymerase gene, but I would rather put it as a separate gene, which is a protease. All right, so the protease is also present inside the virus. Right, now we have covered the 4 main genes, main group of genes that coded for the protein. All right, also in the virus, the virus also coded for other accessory genes as listed here. And each of them have their own functions, and we'll look at it later. All right, so basically this is the structure of HIV virus. So let's look at the genomic organization of HIV. So the HIV genome is actually mRNA. All right, it's actually mRNA. The mRNA, of course, as you know, the mRNA will have cap, you have the non-translated region, then you have the coding gene, then non-translated region or 3' N, and then poly-A tail. All right, so the genome of the virus is mRNA, and when it being reverse transcribed into double-stranded DNA, so this is the arrangement of it. All right, so gene that coded for specific protein, and we also have accessory genes that coded of different protein. Then the front and the back, we have a long terminal repeat. We'll look at this later on in more specific. Excuse me. All right. So again, let's look at this. GAG gene coded for 4 protein matrix, capsid, nucleocapsid, and the P6 that is the one that on the surface of the capsid protein. And then the POR genes, of course, it has the protease, or rather to put it as a separate gene, protease, P10. Then you have reverse transcriptase. Reverse transcriptase have 2 domains. One domain is 51. This 51 is the genes that coded for reverse transcriptase. And what about the additional here? This additional part of the reverse transcriptase domain is actually coded for an enzyme called RNase H. Okay, RNase H, enzyme that digests RNA. So this extra domain is the domain for RNase H. And then we have integrase enzyme that help the integrations of the viral genome into the host integrase. Then surface envelope gene coded for surface glycoprotein as well as transmembrane protein, GP120 and GP41. Okay, so here, like I mentioned, the genome of the virus is actually an mRNA. It has a cap, it has a poly-A tail, and this is the gene. So, and the front and the end here are all those untranslated region, or UTR, untranslated region, 5' untranslated region, 3' untranslated regions. So when the virus undergo reverse transcriptions, reverse transcriptions, it will produce the pro virus, which is double-stranded. Apart from the genes here that are important for the viral protein, you have untranslated regions, which will then be incorporated into the host genome, and in the host nucleus, it will then be transcribed into mRNA and translate into a viral protein, which is something that we will see later on. So listed in the next 2 slides are some of the example of the functions for the virus. Okay, you can look at it, and you might want to come back to these 2 slides when we look at the replications of the virus. It will help you to understand more, why does the virus produce this particular protein, what is the importance of this protein, and you will come back to this slide. The same goes for this slide. All right, it specifically listed all the signs of protein and the function of this protein. All right, so that's it for the first part of these viruses whose life cycle uses a reverse transcriptase. Thank you.